Okay. So again, good afternoon, Jubert and Teresa. And for this session, we're going to talk about RNA interference. And last time we talked about cloning and also animal DNA sequencing. Although we didn't finish um, all the slides, but then, wait lang, tama ba ko? Ay, no. Supposedly, wait lang, wala na ako na, really? Because I thought I have I have posted the video already, so maybe I forgot last time. No, so we actually talk about this. Sorry for that. We talk about cloning and also DNA sequencing. So I will just give you the um, the links of this one, on the animal DNA uh, sequencing and also um, cloning. So for now, so Teresa, na baka last time?
Ma'am.
So we only talk about DNA extraction. Am I correct? And also DNA cloning. Am I correct?
Yes po.
Okay. So ako sang i-review ha. So we did stop create the embryo through impulse of the cell donor. So we have the so-called human reproductive technology bill in 2000 wherein. Okay, so we did stop, na post na yun ako, but we did stop about human cloning, no, this one in this PowerPoint. And we also have another PowerPoint here as additional knowledge on that. But since we are running out of time, I'm so sorry. So we're just going to run through, no, of our discussions. Kasi I hope, kasi June 5 would be our final examination. So actually, just to finish last time, we have discussed about human cloning, the reproductive technology bill. So we there definitely human cloning is, and that which includes replacing nucleus of an embryo with nucleus of another cell, and embryo clone and trading of embryo or the selling of embryos is definitely not allowed. And even in China, no, by the Ministry of Health, they prohibits surrogate mother, no, and reproductive human cloning and donation of embryos or trading of eggs. So kahit magbenta ng mga itlog. So, and recently, last 2002, according to Clone A, they claim that they have given birth to Eve, which is a second birth Dutch lesbian woman early in 2003, and a third to Japanese couple na nag-clone ng kanilang dead son, which was killed in an accident. So this is just a claim anyway. But then they have this one, they have, if you are going to um what do you call that? If you're going to Google their um their company, it's called cloneaid.com, no. And also Korean scientists led by Dr. Wang, they also produced clone human embryos in 2004, which found to be fabricated. And American scientists also claim that they have cloned 14 human embryos and transferred 11 of them into 4 women in 2009. And so on, also UK granted first license for work towards therapeutic cloning, but this time this is therapeutic, so just to help give life, no. And November 2004 in California, um they have budgeted $3 billion, no, for the Institute for Regenerative Medicine, which is also based on embryonic stem cell research. So as for now, this has been considered yung mga partly lang ng mga embryotic, but none of this are allowed, none of talaga total human cloning, a total clone of a specific person's identity, which is definitely like the clone of Dali, so it's prohibited. And so the views of animal and human cloning differ among people with different religious beliefs and religion. So embryo is already considered a living human, no? So it's up to our it's up to our belief. And the use of stem cells for therapeutic cloning is still and although last time it's still in experimental stage, but nowadays it has been proven to be very effective and acceptable already. So the only thing is that it's so expensive, and embryo left over after in vitro fertilization. So is it a violation of natural birth and cloning a beloved one who dies accidentally? Would you consider that one? So it's all against our natural tradition. And cloning for sterile couples, halimbawa, hindi kayo magkaanak. So, they also call that cloning, no? Or in vitro fertilization. Under what circumstances do you want to make a copy of yourself? So, Mary Rose, Teresa, what is your view on that? Hello, Kenneth.
Good afternoon, ma'am.
Yeah. So are you amenable with cloning? Actually, we discussed also this one last time. So, musugot ka, Kenneth, nga nakay ka photocopy sa imong sarili? Or even imuhang anak in the future. Nanda ba kay anak, Kenneth?
Wala pa, ma'am.
So,
Pero yes, ma'am. Sagot ko, ma'am.
kapareho dyan nimo kaayo, as in totally.
Yes, ma'am. Akong bapa og ako, ma'am. Pareho kayo.
Pareha kayo mo.
Clone me do
So anyway, for maybe wala may complication if ever, no, but then maybe imuhang son to be in the future will be also like you, definitely. So if this is only for couple na wala jud sila or dili jud sila magkaanak, no, so because of genetic problems or environmental problems. So if you don't have much more question about cloning, then we will proceed with um DNA sequencing, no, and RNA interference, and also animal genome. So hopefully we can finish really no before. If not naman, so you have guide questions naman to to answer for your quizzes, because by end of June we really need to finish, not end of June, end of May pala, kasi finals na natin sa June 5. Okay. So, we are now on, wala na yung mga pangutana. Naman ko, ako ang nasaag. So, we are now on animal DNA sequencing. Okay? So I just got this from sige, togi ko anan. Slide share, no? Okay. So when we talk about DNA, so this is again the same with um, the previous discussion we had on plant DNA sequencing. So, but for we already have discussed, no, na in terms of cell parts and everything and etc. They are actually we have differences, no, in terms of structural and also components between animals and plants and DNA. So we have to discuss them separately. But then they still follow mga the same na mga like, of course we have the basis of GNA. We all know that the adenine, thymine, cytosine, and guanine. And once again, DNA is a molecule which contain the hereditary material in all living cells and are made up of DNA. And genes consist of the DNA that codes one protein and becomes the genome or the sum total of organisms' DNA. So when we say DNA sequencing again, it means that the process of determining the precise order of nucleotides within a DNA molecule. So it includes the methods or technology that we have mentioned earlier, that is used to determine the order of this ATGC in the strand of DNA. So this is actually just the same exactly. So the sequencing of DNA of molecules began actually as early as 1970s with Maxam Gilbert method and later Sanger method. But these are originally developed in 1975 by Frederick Sanger, and in 1987 applied by system marketed fully automated na nga sequencing machines. So there are methods, several methods. So historically, these two are the main methods, the Maxam-Gilbert method or chemical cleavage method, and the Sanger method or chain termination method. And other methods, sequencing methods are the automated sequencing, the whole genome sequencing, and the next generation sequencing. And in the Maxam Gilbert sequencing method, of course, again, these are developed by Maxam and Gilbert in 1977, and chemical sequencing treatment of DNA with certain chemicals. So the DNA here cuts into fragments, monitoring of sequences. So the principle behind this is the partially cleaved DNA fragment is subjected to 5 separate chemical reactions, each of which is specific for a particular base, and the resulting fragment terminate at the specific base, followed by the high resolution gel electrophoresis. So last time we have seen what is a gel electrophoresis, and detection of the labeled fragments by autoradiography. So these are just the specific methods on how to do it. No, they label DNA at one end of the 32P and copies, which are divided into 4 samples, and each sample is treated with a chemical that specifically destroyed 1 or 2 of the 4 base DNA in the end. So don't be bothered by the specific formula, no. So those are just the actual sequence that you can see in a particular sample DNA. So these are actually the steps in general, no? Preparation of homogeneous, then addition ng 5-phosphate, then cleavage. So there are 4 ways or pathways, and then you do the electrophoresis and radiography. So this is just how does this part here is magnified here. So ito na yung makikita mo na in the sequencing gel. So if you remember last time in plants, we have seen this one, no? Kung ano ang molecule, if they have high or lower molecular mass. So, depende yan kung saan sila magpo-fall. So, sa Sanger method naman this is the most common na approach na ginagamit, invented by Sanger in 1977 and even won the Nobel Prize in 1980. Also known as the chain termination method or the dideoxy method. In this principle, the enzymatic synthesis ng complementary polynucleotide chains is done and followed by the termination at specific nucleotide position and separated by gel capillary electrophoresis and then reading of the DNA sequence. So basically, pareho lang talaga sila ng principle kasi merong cleaving pa din ng or termination. So the requirements are the following, no? That includes yung single-stranded template mo, primer, DNA polymerase, dideoxynucleotide, and so on. So, the method involves denaturation, primer attachment, and extension basis. So, from A, B, and then C, termination, and run through a gel electrophoresis. So that's the procedure actually in the electrophoresis, no, ayan, and then reading is done, and then you will know eventually the DNA sequence, the deduced DNA sequence. So in the automated sequence methods, so in this technique, we use the fluorescence technique. And the fluorescence label DD and TP are used, resulting DNA strands separated in 4 different lane in electrophoresis and detected by fluorescence detectors. So, itong automated is just by using the fluorescence technique. And, so ito na yung steps, no? Step 1, 2, and 3 automated. So this is inside the machine using the fluorescence. So ito yung makikita ninyo. So we don't have to dig deep into that, no? And we also have the so-called the whole genome sequencing methods using shotgun sequencing method. So in the shotgun sequencing, this is also known as the shotgun cloning, wherein this method is used for sequencing long DNA strands or for the whole genome na. And it looks like, the process looks like this in the step A and B. In the next generation sequencing methods naman is based on the principle of Sanger's method, wherein we have many types of NGS, no? Generation, the next generation sequencing. It could be pyro sequencing, Illumina sequencing, or solid sequencing, and many more if actually. So ang advantage versus disadvantage nito is that mas improve yung diagnosis ng disease and also for biopesticides and identifying crime suspects. So sequencing the whole genome of organisms or human genome project, this is mainly used. However, the whole genome cannot be sequenced at once. So kailangan, it will take time and very slow and time consuming. So again, these have application on forensics. So if you know yung mga police or crime natin na mga results are ito yung mga ginagamit ng mga methods, no, to help identify individuals because each individual has a different genetic sequence. So it's more efficient to identify them through this application or DNA sequencing. And also in medicine, these are used to help detect the genes which are linked to various genetic disorders such as the muscular dystrophy. And even in agriculture, the mapping and sequencing of a genome of microorganisms help to make them useful and more productive, no, for crop and food plants, for instance. So that's all for the nucleotide sequencing, no? Do you have any questions or clarifications? Wait now, watch 5. Jubert, do you have any questions?
None so far, ma'am.
Mm-hmm. So, Lisa, this is in animal improvement. So this is actually just a link for you to find out more the application of DNA sequencing in animal improvement. And this is very nice. So I suppose I can open this here, but something's wrong. So if you are going to check on this, kasi dapat naka-sign in ka dapat kung dapat dapat may research gate ka pala kapag if you're going to open the whole text here or view the whole text. So anyway, this is much more specific application of DNA in the animal for the animal improvement. So let's proceed with our RNA interference naman. So if you don't have much more question, no. So I hope we could finish this. So this would be the before mag-end ang ating session. So I hope we could finish this one. So again, what is RNA interference? So we will talk about the different terms like RNA silencing, definition ng RNA interference and its discovery, and the mechanism and applications, especially for therapeutic and other application. So for when you say RNA silencing, there are terms used to describe RNA silencing. Usually there are 3 phenotypical different but mechanistically similar phenomena. So that means that they are different by phenotypes, but with the mechanism is just the same concept. So we have the so-called co-suppression or post-transcriptional gene silencing or PTGS in plants. I know this is very technical terms which are out of our coverage area. So, but the important thing here is that we just have um we just have to try to understand that these things and the functions and the importance of this for the sake of science. Second, we have quelling in fungi and RNA interference in animal kingdoms. So let's learn one by one for each of this. So when you say RNA interference, it's a mechanism that inhibits gene expression at the stage of translation or by hindering the transcription of a specific genes. So it means imong hilabtan ang RNA. So its initial is RNAi, so it I for interference. So you will inhibit the expression or like kung anong characteristic ng specific gene na yan, like na siya pagka black color, no? So at imuha na siyang isuppress para dili siya mag show up, no? So specifically in the stage of translation of proteins. And how? By hindering transcription ng specific genes. So, instead na i-translate nimo siya or i-transcribe into genes, so i-prevent nimo siya. That's what is interference. So, the targets include RNA from viruses and transposons. So, you use the viruses to do this. So this is the process again by which the sRNA silences gene expression or degradation ng mRNA or translation inhibition. And as you can see here, the DNA which has is translated into messenger RNA and into proteins. So before the transcription of this one. So you act here using the interference of RNA. So itong messenger RNA is being blocked in this point, no? So hindi na siya magta-transcribe. So RNA in interference, RNA double-stranded form breaks down the mRNA for a specific gene, thus stopping the production of protein. So ito yung target niya. So why do we need to do this, no? Hilabtanon kita sa mga genetics, no? Sa genes. So, through defense, because of defense mechanism, no? Defense against infection by viruses, for example, or as a defense mechanism to protect against transposons and other insertional elements. No? Yung mga unlikely na ayaw natin na mga genetic na mga problems. And even against viruses, no, like the COVID virus and genome-wide regulation. So the RNA interference plays a role in regulating development and genome maintenance. So to maintain a beautiful genetic makeup or genome, like for 30% of human genome are already regulated nowadays. So can you imagine if all of this genome can be regulated? So that would mean perfection, no? But nobody's perfect. Yun guys sila. So actually, based on this study, there has been a lot of Nobel Prize winners in the study of C. elegans. So field, no, this is a bacteria. From Craig Mello, Andrew Fry, Robert Horvitz, John Solston, Sidney Brenner, and etc. So they found to work in many diverse species, including mice, zebrafish, insects, trypanosomes, and fungi. And how does this RNA work? So uh, in interference, the sRNA and dsRNA with about 21 to 20 NT are being, and this one, 19 to 25 NT, are encoded by non-protein coding genome. And in the RNA-induced silencing complex, they cleave the mRNA. And the enzyme has a dicer which produces 20 to 21 anti-cleavages that initiate RNAi, and drusha, which cleaves base hairpin into the form pre-mRNA, which is later processed by the dicer. So in the sRNA, this shows the small interfering RNAs that have an integral role in the phenomenon of the RNAi, which form post-transcriptional gene silencing. And these fragments bind to the complementary portion of the target mRNA and tag it for degradation. Hence, a single base pair difference between the sRNA template and the target of the mRNA is enough already to block the process. And each strand of this siRNA has the phosphate termini, hydroxy termini, and so on. So this is the generation of small interference RNA. We have A, temporary silencing. Now this is shown here. And the long-term silencing. So these are these specific parts and how they terminate each of the sequence. And the miRNA originate from cap and polydenyl adenine full-length precursors and hairpin precursors. This one as shown on the figure. So we are less than 10 minutes now. Illustration of mRNA processing from in the nucleus and also in the cytoplasm. So from drosha and then exporting out from the nucleus to the cytoplasm in the dicer and the RISC assembly. So the difference between miRNA and siRNA is that they both functions both species is regulation. Uh, Function of both species is regulation of gene expression, and the one of the difference is that in where particularly they originate. And since siRNA originates with dsRNA, and it's most commonly as a response to foreign RNA, usually viral, and is often 100% complementary to the target. While the miRNA originate with the ssRNA that will form a hairpin secondary structure and regulates post-transcriptional gene expression, and is often not 100% complementary to the target. So ito yung main difference nila. And also, the miRNA help regulate gene expression, particularly during induction of the heterochromatin formation that serves to downregulate the genes pre-transcriptionally, no? Or RNA induce the transcriptional silencing, or what we call RITS. So this is the dicer. As you can see, the sRNA with an enzyme to make it siRNA. So that's called dicer. And RNAs, specific ribonuclease, these are the enzyme involved in the initiation ng RNA, which able to digest dsRNA into uniformly sized small RNAs. And in the dicer family proteins, these are ATP-dependent nucleases, and which also RNAs are enzyme that can be act also as a dimer. And so with the loss of dicer, also loss of silencing processing in vitro, and this we call them homologs. They exist in many organisms like the elegans, Drosophila, yeast, and humans. And dicer is an conserved protein. So the dicer has a domain and it's a ribonucleus with 4 distinct domain. So we have the helicase, the RNase 3, the PAS RNase 3, and the DSRBD. No, the amino terminal helicase domain, and the dual RNase, and so on. So the RISC is actually a large um, RNA multiprotein complex that will trigger that mRNA degradation in response to siRNA. And also this involves in unwinding of the double-stranded sRNA by the ATP-independent helicase. And these active components are in endonucleases, which are known as arachidonic proteins, which cleave the target mRNA here strand. So the steps here involved are first RNA interference, which is a biological process, a natural process in which the RNA molecules inhibit the gene expression, typically by causing the destruction ng specific mRNA molecules. And historically, it was known by other names like co-suppression, post-transcriptional gene silencing, and quelling. So all these are just called RNA interference. So in the first step, the DNA is processed into sense and antisense RNAs, no? And then the sRNAs associate with the RNA introduce silencing complex, and they unwind. And the antisense sRNAs act as guides for the RISC to associate with complementary single-stranded mRNAs. So RAC cuts the mRNA in the middle of the paired with the sRNA, and the mRNA is degraded further. So what do we mean by the sense and antisense RNA? So these are the messenger RNA, single-stranded called sense because it results in a gene product protein. So that's why they call sense and antisense. So antisense molecules interact with complementary strands of the nucleic acids, modifying expression of genes here. So the messenger RNA and the antisense RNA will have will will translation blocked. So, ano ang importance ng RNA, no? For infectious diseases, no, para ma-prevent natin sila, so we need to interfere or to stop the expression of their genes. And in the stem cell biology, as I have seen, yung stem cell has been used for therapeutic na, even for humans. And oncology or cancer treatment, and hematology or for blood. No? So since throughout the years by human existence, meron tayong mga genetic problems with blood. So we have 3 minutes left, so I have to be fast. So in hematology, so this include disorders from gene function, loss of gene function, mutant gene, and absent gene function. And also models of disease processes and pharmacologic and genetic therapeutic targets. And also for cancer by targeting oncogenes or dominant mutant oncogenes, amplified oncogenes, and viral oncogenes. And define role signaling molecules of tumor creation. And improvement ng chemotherapy, efficacy, and radiotherapy. And also to regress the tumor through creation ng potentially new mode ng chemotherapy. And in the stem cell biology, the mouse research, for example, knockout tumor suppression gene in mouse embryonic stem cell, since this has been done in mouse research. And observed tumor phenotype and positive correlation between extent of inhibition and severity of the disease. And for a lot of infectious diseases, no, including virus targeting, they inhibit cellular and viral factors of disease, and so on. Inhibit the replication, so hopefully they could do that in COVID. So the goal is to render cells resistant to infectious organisms. So it's for human thing. So we also have the hepatitis C, which already infecting millions of people worldwide. So this through the RNAi treatment control in virus, and also selling genes in HIV virus. So this has been the main aim, no, to silence the main structural protein in the virus. And also respiratory infection that includes caused by virus. And macular degeneration, no, in the leading cause of adult blindness. And also Huntington's disease. And other uses such as studying cell division, testing hypothesis of a gene function, target validation, pathway analysis, gene redundancy, and functional screening. So these are the different uses and advantages of using the RNA interference. So if you have if you still have questions, um, then we have limited time already. So you just message me if you have more questions and clarifications on our about RNA interference. So klaro ba na since we still have time, so we can have a short reaction, no, Teresa or Mary Rose?
Yes, ma'am.
So what did you understand about RNA interference?